Hi everybody, so the first video that I'm hoping that you all will watch is intended to give you a quick refresher on some of the cell biology aspects of bacteria. So what you can see right here on this slide, hopefully you'll remember this from Biology 315, but this is an isolation streak. What you have here is a dense lawn of bacteria. Here's some of it starting to get diluted out, and then you have these individual spots called colonies. Just as a reminder, each one of these is not a single bacterial cell. Each one of these, if you blow it up like this, and then look at it under the microscope, would actually be made up of billions of individual bacterial cells. Okay, so just to give a quick reminder of where bacteria fall in the tree of life and how long they've been existing, we have this image. This is an image from the Brock Biology of Microorganisms textbook. Sorry about that. Just to give an idea, if you look back here about 4 billion years ago, you have the universal common ancestor, the origin of cellular life. Not too much further than that, you have the development of the first bacterial cells. And they have been here ever since, the entire time. So if this is present day, here are humans, and humans have been around for such a short time that on this type of time scale, we don't even make a mark, we don't even measure. And yet bacteria have been around all this time. Just as a reminder, there are 3 standard bacterial cell shapes that you're going to see, and cell shape and the term morphology are not exactly interchangeable, but we can essentially use them that way. The 3 shapes that we want to deal with are coccus, bacillus, and spirillum. Bacillus and rod are used fairly interchangeably. The plural of coccus is cocci. So what these guys look like, just as a reminder, is that any coccus-shaped cell is going to be just a simple circle. A bacillus-shaped cell, as you could guess, is shaped like a rod. So you're going to look at a cell that's got this shape to it. A spirillum cell is any cell that has curves and periodicity to it. So anything that looks kind of like this. It would be bad not to point out that there are cells that have unusual shapes. Some of them are sort of pleomorphic, meaning they don't really have a defined shape, and others have other distinct ones. We're not going to worry too much about those. It is important to point out, though, that many bacterial cells have just sort of a theme or variation on these base shapes. So just to give... Sorry, quick technical difficulties. So just to give a quick point about how these work is that you can have a standard coccus shape, just a circle, and then you can have something that still is a coccus shape, but it's got some appendages sticking out from it. So it's not a strictly defined coccus. You can have something that's rod-shaped and is filamentous. So that would be something like these down here. These are still rods, but they're incredibly long and skinny. They don't have this kind of standard shape. Also, what you can have are variations on spirillum, and it's in particular seen in a class of bacteria called spirochetes, that instead of having just sort of a little curve to them, you have this incredible, like, fusilli pasta looking shape, where they have really, really tight spirals. So just as a reminder of how big bacteria actually are, they are not very big. So you have an established size range of between about 0.2 microns or 200 nanometers to right around 700 microns. Here are some of the biggest, here are some of the smallest. Just to give you some context to that, the size range for a eukaryotic cell is about, right here, 10 to 200 microns. If you look at the bookends here, so these 0.2 microns and 200 microns, you really see that between the smallest bacteria and the largest eukaryotic cells, you've got about a 2,000-fold difference in size. Just to give you a nice image of that, these black specks right here on this image, these are Mycoplasma hypopneumoniae cells. This is a bacterial pathogen of pigs. This surface right here is the surface of one single swine epithelial cell. So if you want to think about it being drawn out, the pig cell, the swine cell, would be yay big around, and the bacterial cells are just these little guys right here. So far, far, far smaller. Bacterial cell structure is pretty simplistic. If you like, it's fairly unstructured. The inside of a bacterial cell is going to have just a few standard things in it. First of all, you're going to have a chromosome. It's going to be one, with a few exceptions that have just recently been discovered, it's going to be one single circular piece of DNA. So if you take this purple stuff right here and you expanded it and uncoiled it, you'd have one chromosome. It would be shaped like a circle, and this would be DNA, so it's going to be helical. What you'd also see inside of every single bacterial cell are these specks right here. These are ribosomes. Just as a reminder, ribosomes make proteins. In a eukaryotic cell, where you would find these is in very tight association with the endoplasmic reticulum, so there's a great deal of organization there. In bacterial cells, it's not like that. They're just kind of floating all over the place in the cytoplasm. So there isn't one spot in this cell where proteins are made. They're made everywhere. Some other things to point out in this cell is that here's the interior part. Right here you have a cell membrane. Around the cell membrane, in most cases, with a few notable exceptions, you have a cell wall. And that's about it in terms of things that are pretty standard on every bacterial cell. There are some things that you might find that are unique to certain species or present in a good deal of bacterial cells, but not all of them. That would be things like flagella. So this guy right here, certainly not unusual to find in bacteria, but not a standard feature across the domain. Same thing with these little spiky guys here. These are called fimbriae. These help in motility. They also help in adherence. Another thing that you might see in some bacterial cells are something called plasmids. These are extra chromosomal pieces of DNA that you might find just floating around here. They might encode a small number of genes. Certainly not as big as a chromosome, and certainly not essential for all bacterial cells to have in order to survive. The last thing you can see on this picture that a lot of cells have, but not all, is this external layer that's outside of the cell wall. This is in this case, this is called a capsule. These are typically made of polysaccharides. They're fairly complex. They're very important in virulence, so we will talk about them again, but they are not, by any stretch of the imagination, uniform across all bacterial cells. Okay, so an important molecule to talk about when you're talking about bacterial cells is peptidoglycan. So this is a very complex carbohydrate molecule. The reason it's important to talk about is that it is basically the entire makeup of a bacterial cell wall. Or at the very least, you can say it's critically important to bacterial cell walls. It's a very stiff carbohydrate, so it's going to give bacterial cells a lot of structure and a lot of rigidity. What you can see is that this here is a single molecule of peptidoglycan, but there's a lot of different components to it. Forgetting the names for a second, if you wanted to just look at these individual balls, you can see that there's a lot of layers of structure to this. You have this individual string of single polysaccharide units that are linked together. It's next to another string that's linked in the same way, and that you can also see that these two strings are linked to each other internally. This, again, entire thing is peptidoglycan, all put together. What these individual balls are two polysaccharides. One of them is called N-acetylglucosamine, sometimes abbreviated as NAG, or in this case, that's what these G's stand for in this diagram. Another is N-acetylmuramic acid, or NAM. That's what the M's stand for in this diagram. It's, I think, worthwhile to point out that we as eukaryotes make this carbohydrate. We do not make this one. So NAM is exclusively found in bacterial cells. It is not found in eukaryotes. Okay, so to get back to this point for a moment, this is the same image that you saw on the previous slide, this big complex view of a peptidoglycan molecule. What I brought up a second ago was that you have these linked polysaccharides. You have a NAM, a NAG, a NAM, a NAG, linked to each other side by side. And what we also brought up a second ago was that you have these blue cross links between them that actually hold this entire thing together. Okay, the way that these linkages work, again, these are, I didn't bring it up yet, but these are called cross bridges. The way that they work is that they really don't have a whole lot to do with the NAG molecules. They have everything to do with the NAM molecules. And specifically, they have to do with this portion right here. This is NAG, this is NAM, by the way. Forgot to point that out, sorry. What I think it's important to notice is that you have this very large side group, and in particular you have this R group and all of these groups that are going to be very good at interacting amongst themselves and hydrogen bonding with one another. When you look at a peptidoglycan molecule drawn this way as opposed to this way, to me it seems a little more intuitive what might be going on here. You have NAM, NAG, NAM, NAG, NAM, NAG, same as you would here, NAM, NAG, NAM, NAG, NAM, NAG. But what you can see in this orientation is that the external group of this NAM molecule is interacting with the external group of this one, and both of them are interacting with the external group of this one. So this is how you have a string of linkages lining up side by side that are then held together by these NAM-NAM interactions. The important point to that is that instead of having a whole bunch of strings of carbohydrates kind of lined up next to each other, they're now actually bonded and held together. That gives an incredible amount of rigidity, again, to this molecule. That's what makes it such a good cell wall component. Okay, so cell walls are really quite important in taxonomic bacterial classification. I'll make a little comment in a moment about how that's different from phylogenetic classification. The way that this is typically done is by something called gram reaction. Hopefully you all remember from introductory micro what a gram-positive versus a gram-negative cell is. Hopefully you all remember again how to do gram staining from the lab from that course. As a quick refresher, gram staining is a differential stain, and certain classes of bacteria called gram-positives are going to turn purple. Certain classes of bacteria called gram-negatives are going to turn pink. If you do not recall this at all from General Micro, please go read the gram stain Wikipedia page or something. Remind yourself what that actually means. Because the point that I want to make is why it's important in bacterial classification. What you see in gram-positive cells, and it has everything to do chemically with why they turn purple. Again, refresh yourself on that stain if you don't remember how it works. Gram-positive cells are unique and distinct from gram-negative cells by having a very thick layer of peptidoglycan. So if inside the cell is down here, if you like, in this model it's right in here, the first thing that you're going to run into is a cell membrane. That would be this blue line right here. On top of it, you're going to have a very, very thick wall of peptidoglycan. So that would be this external bit right here. This is quite different from gram-negative bacteria that have a completely different cell wall. If this is the interior of the cell down here, or right here in this diagram, first part's the same. You run into a cell membrane, a cytoplasmic membrane. It's that blue bit right there again. Right here, you have a very thin layer of peptidoglycan. So you can see on a kind of molecular diagram level, this and this are probably made up of the same thing, but this one's far thicker than this one. The other huge difference is that on top of the peptidoglycan layer here, you have a second cellular membrane, and that usually you have a whole lot of modifications coming off of that. These modifications I'll talk more about in a second, but these are called lipopolysaccharide. Lipopolysaccharide is completely distinct to gram-negative bacteria. You do not see it in gram-positive bacteria. You also do not see it in eukaryotic cells. Gram-positive bacteria have a similar modification. They have something called lipoteichoic acids. You don't see those in gram-negatives. You don't see those in eukaryotes. Completely distinct to gram-positive cells. Okay, so key differences between these two. are that gram-positives have that big thick peptidoglycan layer, and they have, I'll draw it for you for fun, and here's their cell membrane, and they have lipoteichoic acids. I'm going to draw those as triangles. Gram-negatives have really thin peptidoglycan layer. They have a membrane on top, a membrane underneath, and they have lipopolysaccharide. I'm going to draw that as circle modifications. One thing I did not mention on the previous slide, because it's a little hard to appreciate on the diagram, is that there's actual space in here. So you have an inner membrane, an outer membrane, your peptidoglycan layer, and then you actually do have some gaps. This is called periplasmic space. The reason why it's important to note from a cell biological perspective is that if you think about things like membrane transport, things need to come into a cell. Whoops, that's going out of a cell. Things need to go out of a cell and come into a cell. Things need to go out of a cell and come into a cell. If you have to go through a big giant wall and one membrane, you're going to do this process mechanistically different than if you have a teeny wall, a whole bunch of space, and two membranes. So your transporters here and here are going to be quite different than here and here. So the chemistry and the setup of their cell walls are going to really dictate how mechanistically cell transport happens. Okay, so I mentioned a moment ago two molecules, one called lipoteichoic acid that you find in gram-positive cell walls, and another called lipopolysaccharide that you see in gram-negative cell walls. These are great examples of something called pathogen-associated molecular patterns. They're not by any stretch of the imagination the only ones. Another great example is flagellin, which makes up bacterial flagella. There are many that are associated with viruses. There are many that are associated with parasites, but these are two in particular that are really, really well studied. Pathogen-associated molecular patterns are incredibly important during our immune response to infection because they are the very first signal to your immune system that something is attacking you, something is starting to invade you. Okay, so I just grabbed this picture out of my Dropbox folder, and basically what this is a big, giant, busy, scary diagram about how one might start the process of initiating an adaptive immune response. So this right here would be the cytoplasmic membrane of a host or human, mouse, dog, whatever's being infected, immune system cell. Right here on the surface, there's a whole lot going on, but what I want you guys to pay attention to are these bits right here, these things labeled TLR with a number. TLR stands for toll-like receptor. Toll-like receptors, again, are found on host cells. PAMP are found on the surface of pathogens, bacterial, viral, parasitic, whatever. PAMPs associate with toll-like receptors. So imagine you're a host immune cell, you're hanging out, you have your toll-like receptors, and they're just minding their business. There's nothing bound to them at this point. What you can see here, and those of you who've taken cell biology, hopefully you might remember a bit of this, is that when you have a eukaryotic cell receptor and nothing's bound to it, your receptors are in a certain conformation, and there's a whole bunch of stuff stuck to them on the inside. And suddenly, when something binds to them, in this case, you have two TLR4 molecules that something's going to come along and bind to, the shape of the receptor on the inside changes. And when the shape changes on the inside, all of these proteins are suddenly let go, or some of them are suddenly phosphorylated, or some interior signal happens to this cell that lets the components on the inside of the cell know that this receptor has had something stuck to it. Usually something sticking to TLR4 or TLR2 or TLR6 tells this cell that it's now being attacked. Something is invading it, or something is invading the body that it belongs to. And so all of these players on the inside, not to worry about the details, that's what Dr. Snyder's class is for, These guys are going to let go. They're going to start interacting. They're going to start signaling to one another. And then eventually, way down here somewhere, this cell is going to get the message that it needs to start expressing different genes that are going to be involved in mounting an immune response to whatever's attacking it up here. Whatever first signaled through this receptor that there's an attack going on. Okay? Another cool point, that wasn't good. Another cool point about pathogen-associated molecular patterns, if I can just drop that over there for a second, is that they've actually become very useful experimental tools. Hold that thought for a second. Let me grab another picture. Okay, here's my other picture I wanted to grab. This is an image of a macrophage, a human macrophage that somebody grew in a dish. And this person was doing an experiment to examine how Salmonella enterica cells interact with this macrophage. Do they sit on the surface? Do they invade it? When they invade it, are they degraded? What happens? So they wanted to take some pictures with that. They needed a way of somehow specifically labeling the Salmonella cells. Salmonella are gram-negative bacteria, and they have, as all gram-negative bacteria do, LPS LPS molecules on their surface. So what this group of scientists did and published in this paper from last June, or 2 Junes ago, they actually took an antibody that is going to specifically interact with LPS, and they coupled a blue fluorescent dye to it, and so they took their Salmonella, they had them invade these macrophages or interact with these macrophages, and then they exposed their anti-LPS antibody to them. And then what they specifically did was every Salmonella cell suddenly turns fluorescent and blue. The neat part about that is if they then wanted to repeat this experiment with a different type of bacteria, let's say they wanted to repeat it with Klebsiella, that's another gram-negative bacterial cell, they can use this same antibody to label it. So, because both Salmonella and Klebsiella are going to have LPS. So not only are they pretty cool from an immunological standpoint, from understanding a cell biology standpoint, but they actually are pretty darn experimentally useful as well. Okay, so I mentioned a few slides back talking about Gram reaction and cell walls and how that applied to bacterial taxonomy. So taxonomy is this idea of grouping things based on some phenotype. Phylogeny also has to do with grouping organisms, except in this case you're doing it based on genetics. The reason I bring this up is that when you actually look at how bacteria are subdivided based on their gram reaction, gram positive versus gram negative, and then when you actually now start looking at their DNA sequence, it turns out that the taxonomic grouping and the phylogenetic grouping are exactly the same. So all of these guys are gram positives, all of these guys are gram negatives. The point here is that gram reaction and cell wall is very, even though they are phenotypes, they are very closely inherited and organized by phylogeny. Something like cell shape, that is not the case for. There are cocci and rods and spirilla-shaped bacteria all over this tree of life. But gram reaction and cell wall chemistry, very clear division. I would be remiss if I point out, being that my entire research program is based on bacteria called mycoplasma, that there are certain bacteria that do not have a cell wall. It's not that they feel they don't need one. It's not that they somehow have defective copies of those genes. It's that these bacterial cells are obligate parasites, and they've done something called degenerate evolution. They have lived in association with host cells. This would be the surface of a host lung cell, and these are mycoplasma cells right here. They have lived in such close association with vertebrate hosts for so long that they have actually lost their requirement for that phenotype and therefore lost their genes for it. In addition to some bacteria not having any cell walls, there are also some that have atypical cell walls. And the one in particular that's important to bring up is something called an acid-fast cell wall. If you look at this, this would be a cytoplasmic membrane, so the inside of the cell would be down here. Right here you have a peptidoglycan layer, so this actually does have a legitimate cell wall. But what it has on top of it is an extra layer. This is a very it's a very lipophilic layer. It's very waxy, and it doesn't stain particularly well. It's not particularly permeable to a lot of things, so these types of organisms have to have very specific transport pumps to get things in and out of the cell. The good thing from the perspective of the bacterial cell is that it gives them an increased resistance to osmotic stress, to chemical stress, to toxic stress. From the standpoint of these being pathogens, it also gives them a lot of intrinsic resistance to several antibiotics, seeing as this is the type of organism that causes tuberculosis and leprosy. This is kind of a problem. The interesting thing about this as well is that it has to have a completely different biochemical way of sticking proteins in this. It doesn't work the same way as sticking them in a membrane or sticking them in a cell wall. Seeing as these organisms, just like any other pathogen, have to have things like adhesins to help them colonize, have to have things like enzymes to help them degrade and take in carbohydrates. They really did have to have a separate system of these factors to be able to get them to function and transport through this waxy layer. Okay, so the last few slides I just want to talk quickly about bacterial metabolism, and this is hopefully not going to be as awful as this topic tends to be. But I just want to point out one thing, in that metabolism has a very popular connotation that is just simply not biologically relevant. If you define metabolism, this would be the sum total of all chemical reactions that occur in a cell. Our perception of that is we tend to think about, this kind of nonsense that you buy at GNC and all these billions of books. What metabolism is in reality is this precise balance between something called catabolism and something else called anabolism. If you want to think about this in reality and what this means within a cell, this is a metabolic map of every reaction that occurs in the bacterial cell E. coli. So if we blow this up for a minute, if you want to look at a single molecule of glucose, okay, you've got all of these reactions in one specific pathway that starts with a glucose molecule. On the other hand, you also have all of these guys down here that are that are associating with other glucose-related pathways. Then you have the interaction between intermediates here with other metabolic pathways that are, over here that start with a different carbon substrate. And then eventually, when you get all the way to the bottom, you have some outproducts that they themselves then start interacting with other pathways. So the point I'm trying to reach here is that metabolism, if we look back at this on a grand scale, is incredibly complex. It's incredibly dynamic and interactive. And metabolism, sorry, I was doing air quotes, but probably, of course, no one could see that. Metabolism in and of itself is this sum total of what goes in versus what comes out after it goes through this maze of nonsense in here. Okay, so to bring up anabolism for a moment. Anabolism is sort of the inverse of catabolism. Catabolism was taking things and breaking them down into their components. Anabolism is taking those components and building them into a more complex macromolecule that a cell can then use. Okay, so I'm going to drop in a couple of pictures right here. Okay, so here are 3 examples of incredibly important carbon-based macromolecules that a cell has to generate from anabolism. This is adenine. This is a nucleotide. This is phenylalanine. This is an amino acid. This is an acetylmiraminate. Sorry, I tripped over that. But hopefully you remember this from 15 or 20 minutes ago as NAM. This is a carbohydrate. Hopefully you can remember without me telling you what part of the cell that is important to. If not, pause, go refresh yourself. Okay. These guys Who cares about them? They're little individual component parts. Here's why you should care about them. I'm going to take a moment and drop in another image over here in a second. Put those guys over there in the corner, and let me grab another. Okay, so if you look at these three molecules, this is DNA, this is a protein called neuraminidase, and this is peptidoglycan. I know I was a total jerk and said go look it up, and then I showed you the picture. Haha. I'm just trying to be charmingly obnoxious. I hope it worked out. But anyway, you need adenine to build, plus 4 other bases, to build a DNA molecule. You need phenylalanine and a combination of 19 other amino acids to build intact functional proteins. And you need NAM molecules in addition to NAG molecules to build a functional peptidoglycan molecule. You need a functional peptidoglycan molecule to make a cell wall. You need a functional DNA molecule to encode your genes. And you need functional protein molecules to do stuff that yourself as a cell needs to have done. So you can't have this, or this without having these guys right here. Oh, what's going on? Oh, Lord. Okay, as you might be able to tell, I'm a new iPad user, and I'm trying to figure this out. Okay, there you go. So, you cannot have this, or this without starting with this, or this. You cannot get this, or this unless you are, if you are a truly independent bacterial organism, without taking up some carbon-based compounds, breaking them apart by a catabolic pathway, and then reassembling them by an anabolic pathway. And the balance between those two things, collectively, is what we would call metabolism. I would be remiss if I didn't point out that there are certain organisms that steal these from their environment. We like to think of them as parasites. I want to start by talking about catabolism as opposed to anabolism. The point of catabolism has to do, the major take-home of catabolism is that you need to take certain things in and break them down into their component points in order to have the cell then be able to use them for other things that it needs. Catabolism, for me, breaks down into 2 places. That you need to have cells taking in and using energy from their biological system that they're in, their ecosystem that they're in. They need ATP to do cellular processes, and most ATP is not generated de novo. A cell can produce the actual chemical ATP, but it cannot produce the energy associated with it. It has to get that from the biome that it's living in. So many of these catabolic processes where carbon-based compounds are broken down, the point of it is that they're trying to do that in order to generate ATP and do their own cellular processes. The other idea here, the other thing that they can't just make by themselves, are carbon. About 50% of the dry weight of any given bacterial cell is nothing but carbon, and everything else are the modifications of carbon-based compounds, principally. It's an overstatement, there are exceptions, but most of the components of a cell are going to be carbon-based with other little things decorating them. A cell needs to come up with that carbon from somewhere. The point of having carbon-based compounds is, as I'll show you in a second when we talk about anabolism, they're actually incredibly important in doing cellular functions. So it has to be able to grab carbon from somewhere, break it down, and then rebuild it into something that it can use. Okay. Another thing I want to point out that has slight bit to do with metabolism, and certainly has something to do with biosynthetic pathways, is that there are other micronutrients that are very important that a cell takes in. So things like elemental iron and phosphorus and magnesium and calcium and all these other, sulfur, potassium, sodium, all these other guys. But these guys, as a general rule, are used more in very specific pathways. Calcium, for example, is important in forming endospores, which I'll talk about when we're talking about survivability and immune escape. They're also important in formation of cell walls, which I apparently lost my picture of. Things like elemental sulfur are important in making, at the very least, two certain amino acids, one called cysteine, one called methionine, etc., etc. Okay, so the last point I want to leave you with is this question: what does this have to do with virulence? Any of this stuff. What is cell walls, and what is metabolism and anabolism and catabolism? Who cares? This is supposed to be a class about virulence. Actually, they have an incredible amount to do with virulence. On a very simple level, organisms that grow fast have this type of growth rate. If this is a graph, and this is an optical density, this is the number of cells. Or, sorry, this is time. I goofed that up. Organisms that grow faster tend to be more virulent. So there are many studies that show if you perform an infection study with a strain of a certain pathogen, and one strain, strain A, grows at this rate, and another strain, strain B, grows at this rate, here's time, here's number of cells again, this strain, the one that grows faster, is going to invariably be more virulent than the one that grows more slowly. So I would say that is a direct application to virulence. Another thing that is not so intuitive is that polysaccharides are usually cleaved off by a suite of enzymes called glycosidases. So these guys are specifically not designed, but these guys, the role of these guys is that they cleave off individual polysaccharide units or monosaccharide units from much more complex carbohydrates. This is a twofold point. So these polysaccharides or monosaccharides can go directly into biosynthetic pathways. So if I have a glycosidase called glucosidase, and Hanging with Friends game that didn't get finished, sorry. So if I have an enzyme called glucosidase, and it cuts off a glucose molecule from a much more complex structure, which I'll get back to in a second, it cleaves off one single glucose molecule. That glucose molecule can then go up here and be fed into glycolysis, help this grow faster. The other thing that has just happened is if this mess down here that the glucose was cleaved off of, if this is a component of, say, an extracellular matrix protein, what you've now done is damaged the extracellular matrix. This is the kind of connective stuff between two eukaryotic cells, cell 1, cell 2. If you start cleaving sugars off of this, you start damaging the integrity of this matrix. This matrix is rather important, you see, because it holds these two cells together. And when you start holding together a whole bunch of cells, you don't have a whole pile of cells, you have a functional tissue. If you start damaging this integrity, you stop having a functional tissue at some point, and you start having a pile of cells. And a pile of cells does not work real well as a functional tissue. So you could see that by having this type of enzyme, you're not only going to give yourself an edge in virulence because you're damaging tissue over here, you are also giving yourself a growth rate advantage. And as we talked about up here, a growth rate advantage tends to make you more virulent. So this is a twofer, right? This contributes to virulence in two different ways, but the bacteria probably acquired the genes and retained the genes for metabolic function. So that actually has an incredible amount to do with virulence. Another thing that has to do with virulence, let's go back to this point right here for a moment. Extracellular matrix. If you start clipping sugars off of this, and let's say you clip off a sugar, there's a very well-known example about a sugar called hyaluronan, which is usually abbreviated HA. If you start clipping hyaluronan off, it turns out your immune system also recognizes something called DAMPs in addition to PAMPs. This stands for danger-associated molecular pattern. Like we were talking about a few slides back, there are toll-like receptors that recognize microbial patterns and tell your immune system it's about to be attacked. Same deal works for this. We have toll-like receptors that will actually recognize damaged host cell components. One of them that is well studied is fragmented hyaluronan. If you start chopping it to bits, we have a toll-like receptor that recognizes that. So when you have chopped up hyaluronan, even if the bacterial cell that chopped it up is no longer present, if it's cleared by some other mechanism, so long as this fragmented hyaluronan is still hanging around, it's still considered a danger-associated pattern. It's still going to grab onto a toll-like receptor, and it is still going to cause inflammation. Inflammation, for those of you who haven't taken immunology, that's what makes you feel sick. That's what makes you feel crappy. That's what gives you fevers. That's what gives you pain. So even though the pathogen that caused this problem in the first place might have been coughed out or sneezed out or whatever, you're still going to have some bad effects from that. So metabolism, I won't harp on it too much because I'm about to finish talking and be done with this video. Metabolism is actually incredibly closely linked to bacterial virulence.